All right, can you see my screen?
Yep,
Yes.
looks great here.
Okay, can you see the animation?
Yep,
Perfect. Or we should be all set.
great.
Participation, but it's a little bit hard to solicit questions from an online audience, but I hope you've had a chance to at least ponder these questions a little bit. The question is, can white checkmate black? Here's the king highlighted with a red dot. And if that's possible, how? So we'll stick with just with the first problem for reasons of time, but if you're curious, we can go back and address the other two as well. In the first case, it's very tempting to take the D1 rook and advance it up the board next to the king, because of course this position is protected by the white bishop. However, you can't do that because there's a black bishop that's able to take that rook. And so then you've lost your chance at checkmate and you've got to re-choreograph your strategy. So a good solution here is to do that choreography and take the D8, excuse me, D8 rook, move it up the board into the corner, and that then is able to take the black bishop, which is forced to interpose to protect the king. And in so doing, you can move the initial rook up the board, and now you've established checkmate. Okay, so think about that problem and how the choreography and re-choreography goes, and we're going to come back to it, and I'll skip these others. But if you want to solve them, you're welcome to. So our lab works in a few different topics in complex organic chemistry, including a program in metal hydride atom transfer catalysis, the interrogation of natural products, in particular those that are active in the human central nervous system, and then a kind of fundamental problem in organic chemistry, which is how to establish these attached ring motifs. These are kind of like biaryls, where you have two aromatic rings sharing one bond, but in this case, you have the added difficulty of tetrahedral carbons at the bridgeheads and full substitution, which causes steric repulsion in the transition state to form these. So it's a nice general problem that I'm hoping many in the community will begin to tackle. The reason why we work in natural products is it was really threefold. Number one, their unique function and utility. Number two, their unique challenge. And number three, the difficulty of access. Okay, and I'll try to unpack a few of these in the introductory slides. So number one are there unique properties of natural products. So as you can see in this analysis by Waldman, natural products occupy an area of chemical space that differs significantly from that of synthetic compounds. These are the feedstock commercial products you can buy from, say, Sigma Aldrich or Acros, in addition to approved drugs. These in particular are found in the worldwide drug database, for instance. And this is important, this analysis is important because what it shows you is that drugs are in some ways a compromise between the chemical properties that you might want to enter into a living system, plus the chemical properties that you're stuck with in terms of what is commercially available and accessible. So you compromise in this walk from commercial space over to natural product space. Just to be clear here, a chemical space is a parameterization of molecules. So you can use any number of approximately 1,300 parameters, from total number of carbons, total number of atoms, formula weight, polarity, hydrophobic surface area, that sort of thing. And you can plot those on a graph and then tease apart where molecules lie when you have large collections. A similar finding was reported by Peter Erdl at Novartis, and he showed, again, the natural products occupy a region that's distant, that's far away from approved drugs and synthetic compounds. Okay, and why this matters is that one of the key attributes of natural products is that they're complex. Okay, what does complex mean? Well, again, it depends on what parameters you use. And in this beautiful study by Stu Schreiber and Paul Clemens at the Broad, they define in this case complexity by the number of stereogenic carbons over the total number of carbons. And in this case, what they found is that the more complex a small molecule is, the less promiscuous it is. In other words, the more specific it is in binding to one protein target or a small number of protein targets among thousands. In converse, the more simple a small molecule is, the more promiscuous it is, and you're going to get a lot of off-target binding that you may not want, that may show up later in clinical trials, which can be very costly. All right. So in this respect, we like to think about our field of total synthesis as this navigation of chemical space, where we start proximal to us, that is in commercial space, things that we can buy, and then navigate to these remote areas of chemical space embodied by natural products. So each of these colored globes is then a discrete natural product that we're trying to access. And the key is that if you can navigate to these remote areas of chemical space that no one else can navigate to, or better than what other groups navigate to, that means you're poised to discover biology that nobody else can discover. Okay, and in order to do this, oftentimes we have to invent or discover new chemical reactions to accelerate this navigation of chemical space. So in the beginning of the talk, I'll go through, it will be quick, but you can find all this information on our website. I'll go through a few abstracts of how we've done this navigation through chemical space and what we've discovered. We have a longstanding interest in what are called the isocyanoterpenes embodied by these complex structures here. They're very interesting in that the carbon-nitrogen bonds are biosynthesized not from reductive amination and monic reactions, as you often find with alkaloids, but rather it's thought the capture of high-energy carbocations by cyanide. And one of the interesting properties embodied by this class is that they're potently antimalarial. They'll kill the Plasmodium falciparum parasites that cause malaria. And at the time we got involved in this, it was thought that their mechanism of action involved inhibition of heme detoxification, that is crystallization of heme that's released in hemoglobin catabolism, but that only occurs in the blood stage. So in collaboration with Elizabeth Winsler at UCSD, one of the premier researchers in antimalarials, we found that these compounds actually affect antimalarial activity in the liver stage of malaria, where there is no hemoglobin catabolism, meaning that this hypothesis in the literature of heme detoxification was wrong. Now, we don't know what the actual mechanism is yet, but we're working on this in collaboration with a few groups to try to pin it down. In order to navigate quickly to this area, we discovered a kind of unusual reaction that allows the stereochemical inversion of chiral tertiary alcohols to alkylamines. This is a kind of reaction that you learn in introductory to organic chemistry is wrong. Okay, and that's because oftentimes these reactions proceed through an SN1 reaction leading to a planar cation, and those can be attacked from either face. In this case, what we found is that these are likely to proceed through a carbocation, but through what's called a solvent cage pair, where the leaving group, even though it's ionized, has blocked one face, allowing the nucleophile to come from the other face, leading to stereochemical inversion. So we can take bisabolol, for example, and invert the stereocenter to bisabolyl amine, with about 6-to-1 DR relative to the other stereocenter. This is the fastest synthesis of a natural product that I've ever been involved in. The postdoc here was Sergei Pronin, who's now a tenured professor at UC Irvine. He conceived of this synthesis and obtained the proton NMR of the natural product in 2.5 hours. So that's certainly a record in my lab. I'm very proud of that little factoid. Another area that we got involved in early were the Nufar dimers. These are unusual freshwater alkaloids that have this unusual thiospirane structure, a 5-member tetrahydrothiophane. And although in the past it had been shown that the aminium carbon was electrophilic, as you'd expect, nucleophiles add to this carbon, we hypothesized and eventually uncovered evidence for the fact that actually the sulfur can behave electrophilically. And thiols tend to add into the sulfur, break the carbon-sulfur bond, and we're able to then monitor that across the proteome using Ben Cravath's isotope ABP assay to find that the sulfur is electrophilic, releases a highly electrophilic warhead, and then extensively labels proteins. In order to access these natural products, we developed 2 new methods. One is fairly esoteric. That's the asymmetric Bissell difunctionalization of cyclopentenones. And the other one is a general method for the oxidative cyclization of tritylthioether-enamine pairs with iodine. So you oxidize both of these species that collapse to the pharmacophore that's embedded in these natural products and labels the proteome. A longstanding program in our lab was developed from the Illicium terpenes, which have an interesting phenotype of neurite outgrowth. The mechanism, the time we got involved, was not known. We believe that now it causes hyperexcitation of neurons, as shown in collaboration with Eli Lilly, through a specific inhibition of GABA-A receptors. And en route to this series of natural products, we discovered an unusual butenolide heterodimerization. So here are 2 butenolides. They're unsaturated 5-member lactones. We can dimerize them both heteroselectively and stereoselectively to give rise to these attached ring motifs. But one of the things that was unusual about it was the speed with which the reaction takes place. The biomolecular rate constant is greater than 5,000 per molar per second, which is about 1,000 times faster than a lithium enolate aldehyde aldol. And that's unusual, of course, because you're forming fully substituted stereocenters here. So we determined both experimentally in our lab and then supported by computation with Ken Hauk and Shuming Chen, that what happens is that the major aggregate of the nucleophile, this lithium enolate cubic tetramer, undergoes a rate determining monomerization, so deaggregation, assisted by the electrophile that you add, so that the rate determining step turns out to be deaggregation, actually not carbon-carbon bond formation. And the carbon-carbon bond formation is so fast because these complexes, the electrophile and the nucleophile have pi orbitals that are predisposed to react. There's perfect complementarity between the filled orbitals and the empty orbitals of these two species. Another area that will help us transition to the topic today was interrogation of the azmarine alkaloids. These are cytotoxic compounds from sponge found in the Red Sea. We determined that they're cytotoxic by way of sequestration of iron from the labile iron pool in mammalian cells. We showed this by rescue of phenotype with exogenous iron, and there were iron responsive proteins also were inhibited by this small molecule. And en route to this natural product, we discovered some kind of interesting series of reactions that we think involves metal hydride atom transfer catalysis. So I'll just highlight a couple of these. The first one is hydrogen atom transfer hydrogenation. So normally when you hydrogenate alkenes, let's say with a precious metal catalyst, this proceeds by way of a syn addition across the alkene to give you, as expect, now hydrogens on the same face as of the alkene. But in a hydrogen atom transfer catalyzed reaction, what we found is that oftentimes you have anti-addition of the hydrogen across the alkene, and that leads often to the thermodynamically preferred product of hydrogenation, whereas with precious metals, you get the kinetic product. So this has filled kind of a useful gap in hydrogenation chemistry. And then finally, we were able to adapt this to various cross-coupling modes. And probably my favorite example from the series of reactions is a carbon quaternization. So, you learn in undergraduate chemistry that you can take amines, you can form the quaternary ammonium salt with, let's say, alkyl iodides. Well, here now you can take alkenes and form the quaternary carbon, also with simple halides. We do that by dual catalysis using manganese and nickel base metal catalysts. And this area of work has assisted us in the synthesis of what I'll talk about today, the galbulamine alkaloids, kind of interesting series of plant metabolites. They form this small collection of compounds within natural product remote space. They're pretty hard to access via isolation. So I'll tell you about the synthesis of two of them, GB18, an interesting attached ring problem, and himgaline, this fully sp3 alkaloid natural product. These both proceed by way of a key step that involves nickel-catalyzed cross-coupling. Okay, the galbulamine alkaloids, I'm hoping will form the basis for a long program in our lab. They're isolated from this tree, Galbulamima belgraviana. There are two separate populations of the Galbulamima genus in northern Australia and then Papua New Guinea. It's still a little bit unclear whether they are two species or one species of tree. They produce a series of different alkaloids within their bark, the galbulamine alkaloids. Mostly they're called GB alkaloids, but they have some common names like himandrine, himbacine, himgaline. They were isolated actually just after World War II by Webb and co-workers, and their structures were assigned over the course of over 70 years by Ritchie, Taylor, and Lou Mander, who just passed away last year, a very well-known isolation and synthetic chemist from the University of Sydney. So one of the things that all of these natural products have in common is that they have this piperidine core bound to a decalin motif by a 2-carbon linker. And this is essentially what defines the galbulamine alkaloids. This core is synthesized by way, it's thought, of a Diels-Alder reaction from an aminopolyketide. The Diels-Alder reaction forms this unsaturated decalin core, and then a series of monic and oxidative cleavage and carbon-carbon bond formations occur to yield this family. You can check out the work of both Jack Baldwin and Mo Mofasagi at MIT if you're curious about the biosynthesis. But the one area lacking in the characterization of the galbulamine alkaloids is assignment of function, okay? And this is particularly interesting because these compounds are used in traditional Papuan medicine to both affect analgesia, that is treatment of pain, especially for gastrointestinal distress, but as also to induce hallucination in various religious ceremonies. And it's not known what species both cause analgesia and induce hallucination. Analgesia, as you know, is a major topic right now in development of new medicines because of the opioid epidemic and our desire to replace opioids. And hallucination or hallucinogens have become a very important topic with the approval of ketamine, which is a hallucinogen, a dissociative hallucinogen that's been used now to treat resistant depression and led to a reinvestigation of hallucinogens. The difficulty in this class is that it's pretty hard to get your hands on each one of these metabolites to characterize them. They occur in trace quantities in most bark, and sometimes you can't find the same alkaloid from tree to tree. It actually varies depending on what bark sample you look at. Now, there has been interesting work by SmithKline and French, which is a now defunct company that has been evolved into GlaxoSmithKline. They identified one of these compounds, himbicine, as a potent tachycardiac reagent. That means it increases heart rate. And they have now advanced that, as I'll show you, in drug development. It was defined very early on as a muscarinic receptor antagonist by Giolani and Coben at University of Sydney. But this is the only alkaloid within this class of about 40 alkaloids to have a biomolecular target assigned to it. There are a lot of benefits of this class. They appear to be orally bioavailable because they are chewed by humans and they're ingested, and they don't appear to be acutely toxic. They're apparently CNS penetrant based on the observation of hallucination. And they're pretty potent, it appears, because you can chew 1 gram of bark, which contains less than 5 milligrams of a mixture of alkaloids, and this has CNS effects. What, however, is not known is, in particular, are the hallucinogenic and analgesic effects observed actually due to the GB alkaloids or trace other alkaloids that are well characterized as CNS active, like tryptamines, phenethylamines, or beta carbolines. And we don't know that, but we have a good lead, I think, that actually it is the GB alkaloids that are CNS active and psychotropic, and I'll tell you that soon. So one of the compounds that attracted our interest was GB18, and that's because of a hypothesis by an ethnobotanist named Benjamin Thomas, who thought that maybe GB18 was responsible for some of the psychotropic effects of the bark. In particular, he pointed to data from SKF, the SmithKline French, that if you dose this into mice at just 5 mg per kg, so that's about 300 micrograms per mouse, it doesn't affect the pain threshold at the same dose that it inhibits preening. So there's cleaning of a sticky substance off the fur, which to them indicated then that it was not changing the sensation felt by the mouse, but rather the behavior exhibited by the mouse. So therefore it could be mood modifying or behavior modifying and therefore psychotropic. So we were also intrigued not just by that function, but also by the structure. This embodies an attached ring problem. So as I mentioned, biaryls are pretty easy to form by Suzuki reactions, but if you make these tetrahedral bridgeheads, that's hard to control, especially if you want to control stereochemistry. And each attached ring problem is its own problem. It's very hard to generalize. So we thought it would be a great opportunity to solve that problem and potentially come up with a solution that's more broadly applicable. So I'll take you through a couple generations of retrosyntheses and forward syntheses to show you what we came up with. We thought we might address one of the attached rings simply by hydrogenating a pyridine to access this piperidine, where we hoped both hydrogens might come from the same phase, which is a big assumption on our part. And the other retrosynthetic transform that we used was we simplified this unsaturated ester to a ketone. I'll show you why, kind of looking ahead, we did that. So we thought then we could form this remaining bridgehead carbon, new stereocenter here, by way of an arylation reaction. But what we'd have to do is attach this oxygen carbon bond, which you see here, and the carbon-carbon bond to the aryl on the same face. We thought we might use some chemistry from John Wolfe at University of Michigan. That ended up not working. We had to retool a little bit. So that became a real stereochemical problem. The other one was this pyridine hydrogenation. So as you can imagine, this pyridine is freely rotating, okay, which means that the Re and C faces are equally exposed to, let's say, a metal surface, which means control of stereochemistry is difficult. And this is the kind of problem where we thought maybe we could come up with some ways to control it, but really we would cross that bridge when we came to it. The reason we thought both of these challenging problems were at least worth it was because we thought we could easily form this core, by what's called a Danheiser annulation, where an allynyl silane adds conjugatively and then in an alpha alkylation fashion to an unsaturated enone. If you're not familiar with this reaction, it's easy to find Wikipedia, and I'll go over the mechanism soon. All right, so that was our design. Here is our execution. I'm also proud of this little factoid. So Stone Wu, who's the sole co-author on this synthesis, completed everything you see on this slide in 9 months. And this was his second generation approach. I'm not even showing you the first generation approach. That has nothing to do with me. Stone is a real prodigy in the hood. He is Australian from University of Sydney, and I cannot recommend him highly enough. So here we start with the Robinson annulation product, methylvinyl ketone and cyclohexanone. We epoxidize from the convex face so that the epoxide and this hydrogen are on the same face. And then we hydrogenate with palladium on carbon that cleaves the alpha bond to oxygen to give you this beta hydroxy ketone. Then en route to a Segusa oxidation to make the enone, we form the double silyl ether with TMS triflate and triethylamine that selectively gives us the 2,3 enol ether over the 1,2. And then we can affect a stoichiometric Segusa oxidation with palladium acetate and acetonitrile to give us the enone. And now we can experiment with our key Danheiser annulation. We take this allynyl silane and titanium tetrachloride at -78. The first thing that occurs is a conjugate addition to the 4 position of the enone that gives now a silonium stabilized vinyl cation. Is thought to have a structure similar to this, which is rapidly trapped intramolecularly by the titanium enolate, now to give you the 5-membered ring. Very cool reaction, a formula 3+2, and it's perfectly stereoselective because it adds again to the convex phase. So now we tried some of the Wolf chemistry using palladium to do an alkoxy arylation. That did not work, so we retooled a little bit. We treated with the vinyl silane with N-iodosuccinimide that gives you the vinyl iodide, and then a stoichiometric Stille cross coupling gives us now the unsaturated pyridine. We could then affect a halo etherification with ICL and silver carbonate to give you now the very strained oxo bridging bicycle. I'll show you experimentally that it is strained. Never take anyone's word for it when they just say it's strained. Look for experimental evidence. I'll show you that. We could then selectively reduce that benzoic chloride with zinc acetic acid. That comes from the convex face. So we end up with this hydrogen pointing outward. And this is actually a very important observation. This pyridine is now tucked on the endoface of this ring system. It turns out that this is higher in energy than the opposite stereochemistry by about 0.5 kilocalorie per mole. And it turned out that was hard to control in our next generation synthesis. As we feared, hydrogenation of this pyridine to the piperidine was non-stereoselective and delivered equal mixtures of Re and C phase hydrogenation, leading to a precursor to GB18. We didn't go ahead and finish the synthesis at this point because it was so clunky that we figured we would go back and fix all of these problems that are highlighted in red here. There's a lot of stoichiometric metals, mixtures of different products, low yields, and difficult separations. So let me show you now our final generation synthesis and some problems that we solved along the way. So the first thing we did was compress those first 2 steps with a conjugate borylation. We actually don't scale up using this Molander-like conjugate addition. It's very important for us for control of stereochemistry because we're now trying to affect dynamic kinetic resolution. But it was good proof of principle that we could achieve this in 1 step. We can then compress the Segusa oxidation into a very scalable process using a slight variation on these conditions where we use now catalytic palladium acetate. And we use DMSO as solvent. This is useful because it turns out that the double silane is not soluble in DMSO. It actually separates out as a separate layer at 2 molar. We run it very concentrated. When the reaction then goes homogeneous, we know it's done. We can take that whole mixture and dump it onto silica and then filter out the palladium acetate and end up with about 40 grams of this enone. Now we can affect our Danheiser annulation. This is almost the same as the previous generation, except we're going to quench now with HFIP, hexafluoroisopropanol, which is a moderate bronzed acid at about a pKa of 12. But when you mix that with titanium tetrachloride, it becomes a strong bronzed acid and can protonate off the vinyl silane to give us the parent cyclopentene. Okay, so we're just replacing the silane with a proton. The reason we wanted to do that is that we thought we could maybe take a shortcut through all this stoichiometric Stille cross-coupling silliness and go directly to this endopyridine ring. The question is, how would we do that? What we thought we could do is affect a standard iodoetherification with N-iodosuccinimide. HFIP accelerates this and gives us good yields on large scale. And then we would try to affect Weich's type cross-electrophile coupling with a pyridine electrophile and try to control the stereochemistry through inversion of this iodide, because the mechanisms often involve the intermediacy of carbon-centered radicals. However, the first conditions we looked at were photoredox conditions reported by Doyle and McMillan, and these were not successful. That's because this alkyl iodide, it turns out, is quite photosensitive. So you shine blue LEDs on it, you get rapid decomposition. So instead, we turned to more standard conditions from Dan Weich's using insoluble reductants like zinc and manganese, or soluble reductants like TDAE. These, however, were not successful. Zinc and manganese tended to fragment that strained ether ring. Okay, so this is the evidence. Otherwise, you may not get fragmentation. And then TDAE ended up giving us proto-dehalogenation, also not successful. So here's where we took a page from our previous work on manganese-nickel dual catalysis. This is kind of an unusual reaction because, unlike Weich's type cross-coupling, there was no TDAE, zinc, or manganese reductant. The only reductant we had was phenylsilane. This was originally added to form a manganese hydride in situ, which is the catalytic species. However, it turned out that the manganese hydride appears to also reduce the nickel, which is a little bit unusual and was certainly not designed. So we tried these conditions in the cross-coupling, and it turns out they work extremely well. So using manganese and nickel base metal complexes, we can now affect our cross-electrophile coupling with this 2-iodo-picoline to deliver now a mixture of endo and exo products. So good for yield, bad for stereo control, and it wasn't clear how we would solve this. We initially turned to some amidine ligands that Dan Weich's had reported in 2016 to be particularly effective for cross-electrophile coupling. Again, they didn't really budge the DR very much, but when we started screening against some new amidine ligands that had not been reported before, we found that we could increase the DR first from 3 to 1 and then to 10 to 1. And actually, interestingly, the yield also increases as the DR increases. It turns out that the amidine ligand we settled upon is actually an approved drug. It's called Praxidine. It's an anti-inflammatory, and it's really inexpensive as a result of it being so widespread. So now ultimately, we have a pretty inexpensive and effective cross-electrophile coupling to access selectively this endo product where the pyridine is tucked into the more hindered face. That's quite scalable, and then we can address the hydrogenation of the pyridine. What we first have to do is oxidize the pyridine to the pyridine N oxide with MCPBA. That allows us to then fully reduce with rhodium on alumina, and it gives us 6-to-1 DR now, favoring the correct diastereomer en route to GB18. I don't put any mechanism or transition state in here because the honest answer is we don't know why this works. Our working model is that it's a 2-point binding of the N oxide and this oxygen of the ether to the metal surface, but honestly, we don't know. We're able to then condense the ketone with hydrazine and treat with iodine by way of a Barton iodination to give us a vinyl iodide. These two intermediates are pretty unstable, actually, because of E1CB elimination, again, of the strained ether. But we can subject this vinyl iodide to a carbonylate of esterification that finishes the synthesis of GB18. Depending on how you count the steps, because now people are basically just counting steps to get the paper into high journals, we like to give a range depending on your favorite method for counting steps. And it's 8 to 12 steps depending on what you isolate, what you count as starting material, what you telescope into a one-pot process. The point is not how many steps it is, but the fact that it's very practical. In a single pass in this final sequence of cross-electrophile coupling carbonylation, we can pass through 1.1 or 1.2 grams. And we're able to now compile about 2.5 grams of this natural product. Now, it is the racemate, but we found that we could easily separate the two enantiomers by supercritical phase chromatography to give us now NAT GB18 as a single enantiomer and start probing what the mechanism of action might be. So we first turned to PDSP, which is actually a free screening service that is co-administered by UNC under the aegis of Brian Roth and the NIMH, the National Institutes of Mental Health. And I'll be honest, we didn't expect much, but we got extremely lucky. We screened across 45 different receptors using radioligand displacement and found actually some very robust hits from the opioid receptors. And it turns out that mu and kappa opioid receptors are hit very strongly by NATGB18. So this is just a single concentration of 10 micromolar. So then we looked at a dose response curve just to look at ligand displacement. And it turns out that kappa and mu are about an order of magnitude stronger than delta. And in fact, if you look at the function, these function as antagonists with single-digit nanomolar potency. So pretty surprising. So have we now explained the psychotropic properties? Yes and no, okay? Kappa opioid receptor antagonists are actually a pretty hot commodity right now for their role in next generation antidepressants and or anxiolytics. However, it's unlikely that they would cause hallucination, which is what our ultimate goal is. We did, however, find that ENT-GB18, this is a non-natural enantiomer of GB18, was actually a kappa opioid receptor agonist. Now, interestingly, these are known to cause hallucination. However, they're not naturally occurring. So it is certainly not the hallucinogen that's found in the tree bark. Okay, the take-home message here, however, is that we think we can start organizing all of the structures of the GB alkaloids by function. And in particular, we think many of them are likely to be, by analogy to GB18, opioid receptor agonists or antagonists. So now we're starting to screen throughout the GPCRome to identify now new ligands, new chemical space, and start assigning all of the functions observed in humans and small animals to individual receptors. So along those lines, oh yeah, we just got this hit the other day. Actually, we screened an analog from within the synthesis and found that it's highly selective for kappa across 300 GPCRs. There's basically all noise here, but it hits kappa quite strongly. So we're excited about that, and we're looking to advance that hit. Okay, so another member of this class that we thought we'd look at for assigning function is himgaline. This is a slightly different class than himbacine, which is a muscarinic receptor antagonist, and GB18, which is the opioid receptor antagonist. It's been synthesized before, unlike GB18, in this case first by Sam Chocklum-O'Neill, who was an industrial chemist at Schering-Plough, and actually led to the spearheading a program for the development of Vorapaxar, which you can look up. It's an approved drug based on the skeleton of himbacine. And what Chocklum-O'Neill found is that another natural product, GB13, another globulomyma alkaloid, is a direct precursor of himgaline by way of an azo-Michael reaction, so just forming a bond with this carbon, followed by a directed reduction of this ketone to this alcohol. And many people have followed the same route, so there are both completed and formal syntheses of himgaline and GB13 from Mander, Mofusagi, Evans, Sarpong, and Ma. We thought we'd take a slightly different tack here. So as you can see, this is a highly sp3 enriched target. And generally what people do is they, in these syntheses, each methine stereocenter is formed sequentially one after the other. We thought we could do it in all at the same time in a series of reductions where we start with this aromatic precursor, reduce the pyridine, reduce the aromatic benzene nucleus, and get directly to GB13, which then goes directly through another reduction to himgaline. And in so doing, we thought we could significantly increase material throughput and start interrogating the function. So in this case, we're targeting now as our precursor this, what we call a high fraction aromatic intermediate, which simply has this bridging bicycle between two aromatic motifs. So that's our new target. But the question is, in order to do this through class walk en route to himgaline, we're going to have to control the stereochemistry. And that turns out to be maybe not so straightforward. So here's our high fraction aromatic intermediate. We thought the pyridine reduction, unlike GB18, would be quite straightforward because the only face exposed to the pyridine is a convex face. This aromatic ring seems to be occluding the other face, so we thought we could control that stereochemistry well. However, reduction of the benzene ring might be very difficult, and that's because while one methine stereocenter has the hydrogen positioned outward to the convex face, two of the hydrogens are pointed inward to the concave face. How are we going to control that? And we had some guesses, but we didn't really know. One of the things that we had going for us is that those two stereocenters with questionable hydrogens are at epimerizable stereocenters. So we thought maybe if we can epimerize them, we could get to the thermodynamically lowest energy diastereomer. However, if you look at all of this diastereomers we might form, the naturally occurring stereochemistry is not the lowest in energy. Okay, that's actually 15 epi GB13, and that's not what we want to access. However, what we found in the literature is that, as I pointed out, Chocolate-Menil found that GB13 could be converted to hemgylene by this azomycal reaction, okay, followed by reduction. And we thought that if we get isomerized instead, not at GB13, but at this azomycal product, maybe we could form the correct diastereomer, because when you start calculating all the energies of the azomycal product, it turns out that the correct stereochemistry to get to the natural product is the thermodynamic lowest. Of course, the tricky part is you've got to treat this with acid and then epimerize it with base. So is the retramycal going to outcompete the epimerization? We didn't know the answer, but that's what experimentation is for. So the other reason we like this approach is that this skeleton looked very familiar to something that was already in the literature here. This is a report from the Russian Journal of Organic Chemistry from 2002 from Koltunov. And he found that if you took 5-quinolol and treated it with aluminum tribromide saturated with HBr and benzene, you could doubly protonate 5-quinolol with nitrogen and then protonation at the paracarbon to get to this enone cation, which in presence of benzene would undergo first a 1,4 addition and then 1,2 addition to get you the exact skeleton you want en route to hemgylene. So that was appealing. Unfortunately, this reaction doesn't work. And that's for two reasons. Number one, it's not favorable to protonate this pyridine, this quinoline ring, in the presence of the highly electron-rich phenol, okay, unlike benzene. Benzene isn't as easily protonated. This phenol is very readily protonated, so you never actually favor in equilibrium the protonated quinolol. The other thing we had going against us is that when we eventually did finally synthesize the 1,4 addition product, this turned out to be quite labile to acid. When you treated this with bronzed acid or Lewis acid, you would protonate, again, this electron-rich phenolic ring, and then the enol tautomer from the quinoline side would then cleave the new carbon-carbon bond, and you just get starting materials back. So we had to come up with a different way of forging that attached ring bond. Unfortunately, we had a lot of experience in the lab with, again, reversing the preferences between steric versus contrasteric control and forming these difficult to form attached rings. In this case, you get the Markovnikov product that you would of, let's say, a Friedel-Crafts reaction. But here you're not doing it with strong acids like we want to avoid. Instead, you're using transition metal catalysis. So we thought instead of targeting this cation via protonation, we could instead target this radical. And now the radical we could form under conditions that would not cleave the carbon-carbon bond. How do we do that? If we could intercept it with, let's say, nickel catalysis to form that bond. But how do we form this radical? So I sent the student off on a wild goose chase to find every example of formation of beta-keto radicals I could in the literature. And actually, there are a fair number. We settled on this really nice report by Mattei in 2006, where siloxycyclopropanes could be opened with dicyano-naphthalene, which is a dye that then induces an electron transfer in the presence of light, to cleave the cyclopropane, release strain, and give you the beta-keto radical. In this case, from Mattei's case, it undergoes an intramolecular cyclization, a Giese reaction, the 4 position, to give you now this tricycle. We thought we could instead intercept nickel catalysis. And so this would give us essentially the equivalent of Friedel-Crafts reaction. And as in the case of the alkene hydroaerylation, we'd be able to reverse the normal regio control of cyclopropane opening. Normally, if you treat siloxycyclopropanes with, let's say, an aryl halide and palladium, you get the sterically favored path to give you the 5-membered ring. If this undergoes instead a radical reaction, it should give you the cleavage of the endo bond to give you the contrasteric or 6-membered ring. So in this case, we just need to map our product onto the starting material of siloxycyclopropane, as demonstrated by Mattei. And this itself caused kind of a quandary, because normally if you're looking at retrosynthetic analysis, normally you want to go lower and lower in complexity till you get back to commercial starting materials. This looks like we're making something that's actually more complex than the product. We lucked out, however, in that it turns out this azaindinone is actually commercially available or accessible in one step from 1,3-cyclopentane dione. So in this case, it turned out to be quite simplifying. And here's the whole synthesis of, in this case, GB22, which is one of the aromatic GB alkaloids. So you start from the azaindinone, we form the silyl ether with TMS triflate triethylamine, and then a modified Simmons-Smith reaction gives us a siloxycyclopropane. We had 2 generations of conditions to affect cyclopropane opening aerolation either using the Doyle-McMillan iridium system. It turned out to be extremely sensitive to scale and in particular temperature and other impurities. So we eventually went with Zhang's organic photoredox catalysts. These worked very well, were generalizable across a variety of substrates, and in particular scaled very well in the natural product synthesis. So here we're going to affect the attach ring synthesis. The Friedel-Crafts, it turns out, was extremely sensitive to overly strong Lewis acids, but we found that we could take diethyl aluminum chloride and HFIP, heat them together in toluene, and that affected a very clean Friedel-Crafts reaction now to get this high fraction aromatic intermediate. We could hydrogenate that intermediate with rhodium on alumina to give us the piperidine from the pyridine with complete stereo control. And then reductive amination with formaldehyde gave us GB22. The aerolation turned out to be quite generalizable across a variety of motifs. So you make these aromatic attached rings easily. And we're keeping our fingers crossed that this will be generalizable across a few different coupling partners that could lead to other GB alkaloids as well. To get to hymgleine, we simply start with the methyl ether of the same aromatic intermediate. We then subject this to Ben-Kieser reduction conditions. This is like a Birch reduction, but instead of using liquid ammonia, using liquid methylamine. This is a problem because it's very hard to get methylamine in the amidst the methamphetamine crystal meth epidemic. But we had a way of taking the HCl salt of methylamine, freebasing it, turns into a gas, and then condensing it with a cold finger to make a solution of lithium. And then we reduce, it gives us pretty good, about 6-to-1 DR for formation of this enol ether intermediate. We can then hydrolyze the enol ether with HCl, basify with sodium hydroxide, and beautifully gives us GB13, which we did not really count on. It took a fair amount of optimization, but there was also a fair amount of luck. And then Choclomanil and Evans conditions for reductive amination gives us then hymgleine, completing just a 7-step synthesis of this natural product. We have some preliminary data on what the target might be, but we're still awaiting confirmation, so it's not ready for prime time. Okay, so by way of this attach ring synthesis using nickel catalysis, we're able to significantly simplify this walk through chemical space, whereas rather circuitous in the past, now we can simply start from this proximal commercial space in a convergent manner, access these aromatic GB alkaloids, and then just a series of reduction reactions get us to the fully 100% sp3 hymgleine. Okay, so that completes the GB alkaloid story. In the last few minutes, I'm going to tell you another story that's, I would say, conceptually different. So in the case of the galbulimine alkaloids, we're doing this walk through chemical space from commercial products to get to not just one alkaloid, but we hope a variety of alkaloids where we can organize them by GPCR selectivity. These individual natural products are important because remember, there's already biological data in small animals on the alkaloids themselves, and we'd like to tie that to receptor selectivity. Okay, let's go back to this chess analogy. So here's a natural product, and here's an analog of the natural product, okay? There are structural differences between these two. And that means that if you have a synthesis of the natural product itself that's rather lengthy, potentially the analog of the natural product can deliver a more robust synthesis. So what does that look like? Okay, this is kind of abstract. So let's go back to the chess analogy. Also abstract, but I think it'll communicate what I mean. Okay, so here is our original solution from the beginning. We had the rook attack the king. We couldn't do that because the black bishop was blocking the square. So we re-choreographed, and we had the alternative corner rook attack. And then the first rook comes in. So let's look at this alternative problem. Okay, how does it differ from the first? I'll give you a minute to just eyeball the two boards. How is it different? The only difference is that this offending black bishop here is gone. We've deleted it, okay? Of course, this is against the rules in chess. You can't say, hey, opponent, what's that behind you? And then grab their piece, right? I'm sure you've tried, or well, hopefully you haven't tried, but you can't, that's against the rules. Okay, there are no rules in synthesis. Okay? You try to solve the problem as best you can. You're not constrained because it's not a game. You're trying to solve these problems to make enabling science. So in this case, by deleting that rook, we're able to move our, by deleting that bishop, we're able to move that rook up to the king. We don't have to worry about now the bishop taking that rook. Now we've achieved checkmate. Okay. Now you might say, okay, well, we've solved the problem more quickly because we've simplified the board. And you would be right. This problem is simpler and the board is simpler. But that is not a required relationship because let's say we add a pawn, a white pawn in front of this bishop. We're going to still achieve the same solution, but the board is more complex. We've added more information to that board. All right. So the same is true in synthesis. You can make a natural product more complex. You can make it less complex. There's not always a one-to-one correspondence between structural complexity and synthetic difficulty. So let me tell you a short story about where this came from. I came from a work on Salvinorin A. There are very few natural products that kind of make it into the popular knowledge, but Salvinorin A is one of them because it's the active hallucinogen in Salvia divinorum, or salvia, which is a recreational drug. It's a kappa opioid receptor agonist with a pretty high Ki and functional potency of about 2.2 nanomolar, actually down to 0.2 nanomolar, depending on the expression level of the receptor. It's a complex molecule, so the total syntheses stand between 20 to 29 steps. They're not easy, and they're really not within the realm of a rational medicinal chemistry campaign. The other problem with Salvinorin A is that it's chemically unstable. So if you treat it with acid or with base, it will undergo epimerization of this blue hydrogen, where the all-trans framework will kind of counterintuitively isomerize to give you the trans-trans-cis stereochemistry. Okay, and at the time we got involved in this, there was no published driving force for why that epimerization occurred. We identified two potential driving forces. Number one was a diaxial interaction between this methyl and this benzylic hydrogen. And the other one, a little more subtle, was the presence of this lactone oxygen. When you're in the active trans configuration, which confers potency, there's about a 25-degree dihedral angle between the two carbon-oxygen, carbon-carbon bonds. When it epimerizes to the cis lactone, it achieves planarity, and therefore it gives you an energetic driving force, but that epimer is not active or significantly less active. So we thought we could design an analog of Salvinorin A where the methyl group was deleted and did not have that diaxial interaction, and the oxygen is replaced with a methylene, just CH2 here, because there's no planarity driving force behind the cyclohexanone epimerization. Therefore, we should remove both of those driving forces. The question is, and the reason why people often don't change the structure of the natural product, is because if you change the structure, you're likely to change the function. So how do you know going in that you're going to retain the same function of the natural product? And the answer is that you don't, but you can mitigate some of that risk with computation. So in this case, we turn to Siva Katrich, who is a computational chemist at USC, and Ray Stevens, who's one of the pioneers in GPCR structure. And they're able to dock in silico using a computational model, a homology model between mu overlaid onto the kappa sequence. They're able to dock Salvinorin A and our targeted analogs into the binding site, the proposed binding site. And they found that actually our analogs seem to bind with a reasonable binding score. But we didn't know until we tested it. And Laura Bond, who's an outstanding colleague at Scripps, Florida, was able to then go ahead and test it. So we're able to synthesize this analog. Actually, the synthetic burden was much lower, between 10 to 13 steps, depending on the series. It completely removed the problem of epimerization. After 22 days in the presence of DBU, we saw zero epimerization. and maintain the potency of salvinorin A itself at about 3.3 nanomolar. Again, it depends on the expression level of the receptors. So the question now is, have we made this a more tractable synthetic problem simply because we've made it simpler? Have we made it less complex? And the answer is, it's not so obvious. It depends on how you calculate complexity. And at the time, there were not great methods for calculating complexity. So we turned to what's called the Bottcher score, which is just published in 2016 by Thomas Bottcher at University of Constance. We were doing this all by pen and paper, but my great colleague Stefano Forlì stepped in and helped us automate this with a Python script. So now you can just go to his webpage. You can use a QR code if you want. Input the SMILE string, which you can get from ChemDraw, of any small molecule, and it'll give you the molecular bits of information that that small molecule carries. And it turns out that for our analogs of salvinorin A, they do lose some complexity. There are 466 molecular bits compared to 478, but that's only a 3% change. Meanwhile, the synthetic burden has changed by about 50%. So it's a major decrease in how difficult it is to synthesize, and well within the realm of our now rational MedChem campaign. Okay, so this is how we got interested in this area, and then now I'm already over time, but in the very few negative minutes that I have remaining, I'll tell you another story related to another CNS active natural product called picrotoxinin. In this case, we actually increase the complexity of the target, but decrease the synthetic burden by about an order of magnitude when you look at yield. And now we've been able to collaborate with Corteva. We're almost done this collaboration and ready to submit the paper. We're able to show that it not only retains binding to ligand-gated ion channels, actually increases the utility of the natural product and stabilizes the scaffold against epimerization, and now enables massive exploration of the chemical space around the natural product. So I'm going to go very quickly. I'll basically skip these slides about the origin of this program. It came from our interest in the Illicium terpenes, which have neuric outgrowth phenotype. We're able to synthesize a large amount of one of the representative members, which is now used actually as a positive control in these assays, and show that these are hyperexcitatory agents. They're selective for GABA A receptors, and we think that this correlates well with known models for trophic effects that cause neuric outgrowth. Okay, so the target GABA A receptor is a fascinating target. It's widespread throughout all of life, including archaea, bacteria, and mammals, as well as insects. The insect receptor is RDL, and that's targeted by an old insecticide called dieldrin, which is actually quite toxic to human beings. Both RDL and gluciel are ligand-gated ion channels targeted by ivermectin, which you may know for the Nobel Prize given to William Campbell for its development. It's also been in the news lately. Interestingly, ivermectin is quite safe in human beings, unless you're prone to permeability of your blood-brain barrier, because it turns out that it's actually not very selective for invertebrate receptors of GABA A receptors. So if it does get into your brain, it's incredibly toxic. And so if you're taking drugs that cause that permeability, or if you're genetically disposed, it's a problem. Also, if you're a turtle, it's a real problem. It's very toxic for turtles because it gets into their brain. Okay, so another member of this family that targets GABA A receptors is picrotoxinin, which kind of acts like a skeleton key. It really doesn't distinguish between many of these ligand-gated ion channels, which is good because then you can use synthesis to essentially change the structure and program in selectivity. However, it's quite unstable to base by cleavage of the C15 lactone with acid or with alkali methanol. If you open up this lactone, the cyclohexane can relax and the hydroxyl can then add to an epoxide, relieving strain and irreversibly then decomposing picrotoxinin. There have been, as a result, no analogs prepared by total synthesis. So this is a real challenge that I gave to Stephen Crossley, an outstanding graduate student who's now with Chris Chang at Berkeley, and Mike Lambrecht, who's now at Genentech. So I'll skip over, for reasons of time, a lot of the prior syntheses, but what this illustrates is that all of the syntheses encode the oxidation state by oxidation of alkenes embedded in the carbon skeleton. However, this is very idealistic to do it from a simple parent triene, because you've now passed this point of no return of lactone decomposition. And if you get to the oxidized product from these alkenes, you actually can't lactonize to this very high energy and strained picrotoxinin scaffold. And this is illustrated quite nicely, actually, by some beautiful work from Mike Crochet and Barry Trost, where they attempt to dihydroxylate this complex intermediate with osmium tetroxide. But when they do so, they end up with the wrong lactone, the 2,2,2 bicycle, not the 3,2,1 bicycle that you want for picrotoxinin. So our approach was very simple. We took all the lessons learned from the prior syntheses, especially that from Crochet and Trost, and affected that towards our synthesis, where we would oxidize this diene. But the key here is that we would have no ester in place. On this side, we'd mask it as a methyl group so that the hydroxide that we newly form is forced to lactonize on the remaining ester, and then we can use the hydroxyl that's still free to affect a CH oxidation of this methyl and get to the lactone. So the question now is one of stereo control. How do we control the stereochemistry in a key aldol reaction in the very beginning to establish the right stereochemistry to then progress all the way to picrotoxinin? And that was not so obvious, but I always tell my group, if you're going to fail fast. We failed immediately, actually. When we start with methyl carvone and try to affect an aldol reaction, it actually gives us exactly the wrong diastereomer, 12-to-1 DR for formation of the new carbon-carbon bond, where the incoming electrophile is now trans to this methyl group that we're planning to oxidize. And of course, that's opposite to the cis stereochemistry that we want. The reason we think this occurs is that the stereochemistry is dominated by this beta hydrogen, which does not allow access to the top face. So instead, we want to bulk up the bottom face so that that occludes the bottom face and we force access to the top face, and then hopefully are able to access the cis stereochemistry. However, our initial strategies to do this with a blocking group at this alpha position failed because every time we formed the extended enolate, this aromatized to give us the phenol, because the X's we were using were good leaving groups. So we thought that we could circumvent this by instead symmetrizing to this methyl. Now we don't have to control the stereochemistry of this new stereocenter. And in addition to that, the methyl group is not going to leave as a leaving group. It's a strong carbon-carbon bond. Technically, this is commercially available, dimethyl carvone. In reality, we do synthesize it. But I think what's most attractive about the strategy were the hidden benefits afforded by addition of this extra methyl group. What I mean is that when you look at addition of methyl itself to picrotoxinin, you actually don't increase the volume very much, and therefore you may be able to maintain binding to the GABA A receptor or a similar ligand-gated ion channels, and then not have to go through this lengthy process of excision of this blocking group. So we turned to Tim Carpenter, a great computational chemist at Lawrence Livermore National Lab. He found that methyl picrotoxinin and picrotoxinin dock similarly to the GABA A receptor, give us enough confidence to go ahead and synthesize it. But in addition to that, we thought we might be able to stabilize the scaffold against this decomposition to the point of no return, because when you replace the alpha hydrogen with an alpha methyl group, you might either occlude access to the antibonding orbital of the lactone and therefore kinetically block it from decomposition, or potentially you could affect the conformational lability of the cyclohexane ring open product, because when you replace the hydrogen with a methyl group, in order to ring flip, the hydrogen and the isopropene have to eclipse one another. If that hydrogen is replaced with a methyl group, that eclipsing interaction is very high in energy, and potentially then you can't decompose that intermediate. So we thought there are all sorts of things going for 5-methyl picrotoxinin over picrotoxinin. It would be more stable, potentially it could be as active. Because we could access it easily, we might be able to diversify it and potentially then surpass nature, surpass the natural product, and design this what we call supernatural product, a little tongue-in-cheek because its function may be superior. I loved this idea. My students hated this idea because it's very hard to get a job if you don't make your natural product, which is, of course, this traditional goal of natural product synthesis. But I thought this could be even better than a natural product because we're able to now ask a question and answer it through synthesis, get to new chemical matter that might have better properties. This did not go over well, but we fortunately were very diplomatic in the lab and we decided to compromise, which worked well because it turns out that the natural product and the analog were actually fellow travelers along 5 steps of the synthesis. So we could explore both. we start with dimethyl carvone effect. Now, extended enolate aldol reaction with sodium HMDS of magnesium chloride and 2-octobutyrate. This beautifully gives us the correct diastereo selectivity for the alpha position. The tertiary alcohol stereochemistry is only 3-to-1, but that doesn't matter because we're going to eliminate it in the next step with SOCl2 and DMAP, and then affect a second intramolecular aldol reaction with LDA from now this methyl group. That gives us the 5-6 ring system. We lock the conformation with an old trick from the isolation literature through the bromonium ion, which forms the bromo ether, epoxidized from the outer face with MCPBA, and now affect a dihydroxylation with osmium tetroxide to give us now the lactone, as we hoped, where now one of the newly formed carbon-oxygen bonds, or hydroxyls, lactonizes and prevents its later formation of the wrong lactone. So now, outstanding postdoc Duong Hu Tong took over on this project. He was able to access this intermediate in only 6 steps and really outstanding yields. We were able to make about 2.5 grams of this intermediate and explore its elaboration to the natural product picrotoxinin and the analog. For reasons of time, I'm going to go through this very quickly. It's a series of Suarez reactions. If you're not familiar with the Suarez reaction, it's quite useful actually for CH oxidation. You form the O-centered radical, usually through a formation of a hypohalite ether. The oxygen-centered radical abstracts a hydrogen to form a lower in energy carbon-centered radical, which then intercepts iodine. The iodine then forms, the alkyl iodide then forms an ether with the oxygen to give you the ether. We oxidize the ether with TFDO through a CH oxidation to the lactol. The lactol then decomposes in another Suarez reaction to the tert-alkyl iodide and formate pair. We then reduce the iodide with AIBN 10 hydride from the outer face to give us the axial methyl group, which is poised for another Suarez reaction to give us the lactone. We then deprotect the bromo ether with zinc ethanol, and that gives us picrotoxinin. Is a lengthy and low yielding sequence in contrast to 5-methyl picrotoxinin, where we simply do 1 Suarez reaction, deprotect, and we're there. Okay, so much more robust route. So what is the value then of 5-methyl picrotoxinin? Well, it turns out that we still retain binding to the GABA-A receptor as we hope. We do lose, I would say, a fair amount of affinity. It's about an order of magnitude. Picrotoxinin has an IC50 of 0.2. 5-methyl picrotoxinin has an IC50 of 2.3. However, we realized in retrospect that maybe we could turn these lemons into lemonade if we use this slight loss of affinity to start exploring selectivity against other ligand-gated ion channels. One of them is RDL, and fortunately, I had a contact of Corteva AgriScience, the outstanding Zach Buchan. He worked with Jerry Watson and Ken Beavers and Sam Griffith in electrophysiology, and we're able to make a variety of different picrotoxinin analogs and now look at their varying affinity to the GABA-A receptor in mammals and the RDL receptor in insects. So this graph essentially reflects the selectivity of different analogs to those different receptors. On the y-axis is potency against GABA, x-axis is potency against RDL. We plot our library against these 2 axes. Picrotoxinin in red here, you see is not very selective and much more potent at GABA-A, which is why it's toxic. But we can use our analogs of the 5-methyl picrotoxinin to walk all the way down to this region that's quite RDL select. Now we've lost all this potency, 3 orders of magnitude at GABA-A, but retained all the same potency of picrotoxinin at RDL. And just 2 hours ago, I got the sweet data from an outstanding computational chemist, Avery Sater at Corteva, where he showed that his computational models essentially give us qualitatively that same kind of effect where the analogs of 5-methyl lose potency at GABA, but retain affinity at RDL. And now we're building computational models to understand what is actually going on in the binding pocket to explain this. Okay, finally, we're able to look at stability. As I mentioned, picrotoxinin is quite unstable to base and acid. With just 5 mol% sodium methoxide and methanol, it'll decompose to a mixture of methyl picrotoxate and dimethyl picrotoxinin dicarboxylate. We monitored this by NMR to look at its decomposition, which is complete in 6 hours. To compare the stability of 5-methyl picrotoxinin, we actually did an experiment where we mixed them 1-to-1 because we wanted to make sure that if there was a subtle difference in rate, they were both exposed to the same concentration of methoxide. But what we saw was very surprising. If you treat these under the same conditions in the same NMR tube over 6 hours, here's what you see. Decomposition only of picrotoxin. 5-methylpicrotoxin is 100% stable, okay? Picrotoxin is gone. There's a slight negative slope here, but that's actually error in integration because we can go up to 2 equivalents of sodium methoxide. No decomposition of 5-methylpicrotoxin. And in fact, we've gone out to 109 days, and only then do we see some decomposition of 5-methyl. So significantly more stable, and it makes me wonder whether we should consider picrotoxin a good target for synthesis anymore. After all, it's already made by nature. You can extract it if you want. It's quite unstable, and it's not selective. Through analogs, we can increase stability and make the analog selective. So there's really interesting analysis retrosynthetically that could go into this. I'll let Drake say it for me. I think picrotoxin is really the way to go to start exploring affinity for ligand-gated ion channels. This exploration of what we call dynamic synthetic analysis, we hope to be general. And if you're interested in this philosophy, you can read about it in this Accounts of Chemical Research. So thank you for letting me go way over time. I kind of relaxed and took my time a little bit, but I appreciate your patience. I had to thank my group members. It's wonderful to work with each and every one of them. I really have to thank Stone Wu and Eleanor Landwehr, who are the current group members working on the galbulamine alkaloids. Megan Baker is now a graduated student who's with Merck, who did an outstanding job getting this project across the finish line. Guanghu Tong is now at MIT, an outstanding synthetic chemist who did the lion's share of the analog work of picrotoxin, and of course, all these organizations for collaboration and funding. And you for your kind attention. Thank you very much.
Thank you. Thank you, Ryan. That was really interesting. Going over is not a problem at all. It was fascinating.
I appreciate it.
I love that you have group pumpkin carving. I'm jealous of that.
That has been a yearly tradition now.
Oh, that's a great idea. I might steal that. I will open it up if there are any questions. If anyone wants to jump in.
Yeah, I have a question. So I guess with your guys' success with making, I guess, unnatural analogs of the picrotoxin and salvinorin, I don't remember the slide, but when you were showing the products of your nickel catalyzed reaction for the hemoglobin, you guys made a whole bunch of analogs of that material. Have you guys thought about potentially finishing the synthesis with those analogs and then screening those to see how those bind to different receptors?
Yes, that's an outstanding question. And the short answer is yes. Yes, we're doing that. The longer answer is that we're prioritizing in the hemoglobin route some of the natural products of that series because many of them have already been screened in animals or animal tissue. And so I think the most urgent question that we'd like to answer is what are the receptors in that class? But on the GB18 family, where we have a different nickel catalyzed cross coupling, but it's also general. In that case, we are targeting some unnatural analogs because I showed you very briefly in the bottom of one slide, they've shown some very interesting selectivity across the GPCR.
Okay. Cool. Yeah.
So I'm really intrigued by the chemical space plots that you're showing and just kind of that approach to thinking about molecular design. So I'm going to get selfish with my question here. So my group is a theoretical materials chemistry group, and we are not working on similar targets at all, but we have really huge chemical spaces that we're trying to explore. And there are so many, like you said at the beginning of your talk, there's so many ways to think about comparing one molecule to another one. There's many ways to do that as there are molecules out there. There are tens of thousands of properties you could grab, from very simple to very complex. And it's not ever clear what set of properties is actually getting you close to assessing the types of similarities that are really important for seeing properties emerge and relationships emerge. I don't know if I'm getting somewhere meaningful with this, but I'm curious to try and boil it down to like one thing. How is it that you go about defining your chemical space in a way that is useful for understanding similarities between molecules and targets that you're interested in?
Yeah, so that's not saying the question. I'm glad you framed it that way in terms of defining your chemical space, because there are two ways in which people use chemical space. One, sort of in a universal term where it refers to all of the molecules that might exist given all the atoms in the periodic table. And that's not what we mean. What we mean by a chemical space is defining the parameters by which you evaluate the properties of a small molecule. So I would say the starting point is knowing what your goals are, because if you don't know what your goals are, you don't know what you're optimizing for. Number one, you can't build your chemical space. And number two, you don't know what you're going to change with your target structure, because you can change anything, right? So with respect to material science, not my wheelhouse, so I can't speak to that specifically, but I could give you a great reference. There's, I don't know if you know the name Marshall Brennan. He was one of the pioneers of mediating ChemArchive, which is now sort of taken off as an alternative source for archival science. He has now moved into the private sector and is working with a company called Gabotix, who is working with Alan Asper-Kusik to design new materials. And they're trying to do a closed loop design of computation, perturbation, automation to target functional materials with AI and automation. So while I can't speak to your question for materials, I think if you're interested in a similar way of thinking, that group is a great one to look at. You probably knew that already, though. But for the audience, they know.
I see the distinction you're drawing, but it still feels similar, and like you have so much information that comes out of all of the characterization that you could do, and whittling that down to, the variables that hopefully overlap the most with the story that you want. I don't know. It seems similar to me in that way.
Yeah, no, it is similar. It is similar. So I would say in many ways, it's in my naive way of thinking, since I'm not in materials, I think it's actually easier on our side. On our side, you have, if you have a target that's a receptor, you have the constraints of the receptor. Right?
Yeah.
You can't make your target infinitely large. Is not going to fit. You can get some flexibility in the dynamics of the protein, but you may prevent it from accessing the binding pocket. There are certain motifs you'll probably want to avoid for stability. So unstable intermediates or unstable targets you want to avoid. So you start, like you said, whittling down what you're actually able to modify. And we are in particular interested in going through this exercise with natural products that are unstable. And that again gives you a good reference point and a good constraint on what you can change and what you know and what you need and what you need to change. But I think that's a great point, because it's very easy to get carried away and pie in the sky. It's more important to stay grounded and target the right goals.
Yeah, I feel like I go back and forth all the time.
No, I get the feeling.
Well, are there any other questions? I know we're a few minutes over and people are ready to go.
I've got maybe one, I don't know. To the best of my knowledge, Dr. Shenvi, you're the only one who's really, that I've seen at least, making like unnatural analogs. Do you think that has the potential to become a much bigger field, like kind of like what they do in medicinal chemistry, where they make a ton of analogs and screen them all? Do you think that people could potentially start taking natural products, making slight changes to them and screening all of those? Because I know sometimes natural products have the issue where maybe they're like super unselective, kind of like you talked about. So maybe making small modifications can help them become much more selective for, maybe cancer cells versus human cells or something like that.
Yeah, thanks. This is a great opportunity actually to clarify. So, as long as there have been natural products isolated with individual functions, there have been chemists who want to modify them and make analogs. So by making natural product analogs, in itself, we are not doing anything new. Industry, academia, this has been done for about 100 years. There are great examples of this. I don't even know where to start. Probably some of the earliest work for Sir Robert Robinson. If you don't know his synthesis of troponone, this is a classic example of not only biomimetic synthesis, in fact, it stimulated the hypothesis for the biosynthesis of all alkaloids as involving monic reactions. But it also laid the basis for making unnatural analogs of tropanones, which include, cocaine as a family member, since they were.
Okay.
In fact, believes Robert Robinson's father was a surgeon and very familiar with necessities on the battlefield for treating pain. So natural product analogs are well established, but my favorite example is from my own department. Actually, Dale Boger, who's just across the atrium, has been working on vancomycin for years. If you're interested in the most beautiful examples of subtle structural changes to make natural product analogs, look at his work in vancomycin and how they change a key internal amide bond to an amidine to restore sensitivity of resistant bacteria. Anyway, so that itself, and in fact, probably the biggest inspiration for our work comes from Paul Wender, who's at Stanford, and what he calls function-oriented synthesis, where if you have a natural product with a defined target and a defined binding mode in particular, you can take the key contacts made in that pocket and essentially hop to simpler scaffolds to come up with a much more robust synthesis where you decrease the synthetic burden. What we're trying to do that is just sort of the next logical step in that progression is to focus on the chemical space, because if you scaffold hop from let's say a natural product scaffold to something that looks more like a drug, you are able to do much more chemistry, but what you've lost are the same physical chemical properties that you started with of the natural product, which are important because they often, on average, have higher solubility, higher specificity, again, on average, than simpler compounds. So the only thing that we're doing different is by this exercise of dynamic analysis, focusing on the chemical space embodied by the natural product. But I think that's a great question because it was a great opportunity to clarify the history of this area.
Thank you. Yeah.
Well, Ryan, I'll thank you one more time, and I think it's a good time to sign off. Yeah, really, thank you one more time. It was really interesting, and it was great getting to meet you, even if it's only half of a meeting since it's virtual. But yeah,
hopefully we'll come out sometime if my family ever gets to Flagstaff again. It's almost become a tradition. Maybe I'll stop by the department to say hi.
Absolutely, yeah. Please reach out when you come through town next. That would be wonderful. It'd be great to get to show you around a little bit.
Fantastic. Well, thanks here.
Thank you.
Thanks,
Zach.
Zach.
See you soon. Bye.